Thank you for joining us today for our Genomics of Beer webinar with Dr. Tammy Joska. Dr. Joska has her PhD in Molecular and Cellular Biology from Dartmouth College, where she studied structure-based drug design and patented a group of lead molecules against anthrax. She has published 18 manuscripts in the areas of cardiovascular research, X-ray crystallography, chromatin remodeling, and circadian rhythms. Dr. Joska has been working with GeneWiz for over 3 years. She's a scientific innovator in the areas of gene editing and high-throughput cloning. Without further ado, here is Dr. Joska explaining the genomics of beer.
Thank you for the introduction. Good afternoon, and thank you everyone for joining me for the Genomics of Beer webinar. I'm not only a scientist, I also enjoy brewing beer. There's a myriad of craft beer flavors and character that suit almost any palate. A variety of craft beer is contributed in part by genomics, but by and large today by trial and error. Now and in the future, the genomics of beer may help brewers to craft more precise varieties. After today, I hope that some of you will never think of beer in the same way. I'll start the presentation giving a brief history of beer. This will lead into beer brewing basics, and then I'll talk a little bit about making a better beer. This will lead into the genomics of beer. I will highlight one specific case study, and then we'll talk about the future of genomics in beer. It's quite promising. We'll also talk about resources in the brewing industry. The craft of brewing can be traced back to ancient times as perhaps one of humanity's first biochemistry experiments. Fermentation of beverages may date back to 9500 BC, when cereal grains were first farmed. Archaeologists presume that beer was instrumental in the formation of civilizations. About 5,000 years ago, workers in Iraq were paid by employers in rations of beer. During the building of the Great Pyramids in Giza, Egypt, each worker was paid daily rations of beer, and this served as nutrition and refreshment. In 2004, archaeologists placed the discovery of the world's very first fermented beverage as a mixture of fruit and grain, as well as honey and rice, in Jihua, China. Shortly thereafter, the first barley brew was coined in Mesopotamia. This is modern-day Iraq. Later, Babylonian women drank beer out of straws, so they invented straws to drink beer. At that time, many prescriptions were actually given to people for beer. The first evidence of hops occurred in 900 to 800 BC. Interestingly, up until the early 1800s, beer was thought to be comprised of only barley, hops, and water. It wasn't until the 1860s when Louis Pasteur proclaimed that live yeast are responsible for fermentation. The first pure culture of yeast emerged shortly thereafter in 1883. So in 1150, hops was used initially as a preservative. In 1489, the very first brewing guild was established in Germany. It's now known as Beck's beer. In 1587, the first beer was established in the New World in Virginia. While Prohibition was short-lived, ending in 1933, home brewing was not fully legal until 2004 in the U.S. In fact, home brewing of beer with alcohol content greater than 0.5% was illegal until 1978, when Jimmy Carter signed a bill lifting the restrictions. Today, beer is the oldest and most consumed alcoholic beverage in the world. Beer ranks as third most popular drink after water and tea. Shown here are the number of craft breweries per 100,000 people. 27 of the country's 57 largest craft breweries are in California, Oregon, New England, and the Mid-Atlantic. Oregon and Vermont have 7 to 9 craft breweries per 100,000 people. Next, I'm going to move on to talk about craft brewing basics. The basic ingredients of beer are barley, hops, water, and yeast. Grains also include wheat, rye, corn, and even oats. During the brewing process, fermentation of starch sugars are converted into CO2 and alcohol by yeast. Shown here is a 10-pound bag of Bryce 2-row malt, Mutton's crystal salt, and Dingeman's biscuit malt. These ingredients are used to brew Obama's White House Honey Ale. This is the beer that I brewed to document this presentation. The grains are usually malted barley, shown here on the left, and it's used to create the wort by mixing with hot water through a process called mashing in a mash tun. Shown here is a hot liquor tank. It's heated prior to adding the grains. The hot water or liquor is mixed with the crushed malt or grist, usually in a mash tun. The mashing takes about an hour, whereby the starches are converted to simple sugars that are easily metabolizable by the yeast. And at this stage, this mashing malt can actually boil over, so take caution. The sweet wort or sparge water is drained to remove the grain solids in a process called wort separation. And then the wort is transferred to a boiling kettle. Shown here is the boiling kettle. The boiling occurs for about an hour to denature the residual enzymes from the mashing stage. On the left, you can see the solids that are left over. The wort solids are recycled and used as bird feed or just put into a compost pile. Hops are added during the boiling process to emit aroma and flavor. The longer you boil, the stronger the hop flavor. Prior to cooling, additional sugars and hops may be added. The White House Honey Ale includes different hop varieties and, of course, honey. After boiling, the hopped wort is cooled and the yeast is added. Fermentation may include 2 stages, primary and secondary, depending on the beer. And if you add more hops, the ABU, or the arbitrary bitterness units, will be increased. After fermentation and maturation, the beer is packaged and ready to drink. Shown here are a couple of IPAs. Now we'll move into crafting a better beer. I'm going to talk about beer in general, and then we're going to go into the genomics. Crafting a beer based on character, and then with genomics. Beer taste is contributed primarily by the yeast, but also from various grains and sugars that are incorporated into the flavor as well, like fruits and herbs, and also bacteria. Yeast varieties provide sweet or sour flavor to the beer. They also provide hundreds of aroma compounds that I'll discuss in the genomics section. There are 2 types of beer yeast. There's ale yeast that are the top fermenting type of Saccharomyces cerevisiae, and then lager-style beers include the bottom fermenting types of yeast, such as Saccharomyces pastorianus, coined by Louis Pasteur in 1870. Grains can emit a candy-like sweetness, coffee, or even roasted nut flavor. The hop flower balances out the sweetness by adding a bouquet of flavors with a bitter touch. Hop flavors may include spicy, citrus, floral, evergreen, or even grassy. Some may even include bacon, beans, or even oysters in their brew. Now, it's very important to consider how do you rate a beer? Like how can you tell whether a beer is really well crafted or not? So the beers are rated by appearance. How tantalizing is the beverage? How does it look? Does it make you want to drink it? Appeal also includes the head size, longevity of the head, color, clarity, and carbonation. Aroma and taste before, during, and after effects are considered as well. Palate refers to the overall feel of the beer in the mouth during consumption. Shown here is Heady Topper. It's a double IPA out of the Alchemist Brewery in Stowe, Vermont, and it's rated within the top 50 brews by ratebeer.com. Nomegang is a Belgian blonde brewed with Le Chouffe yeast and bottle conditioned with Omegang yeast. This is a limited edition beer that was created in a bourbon cask. Recently, genomics has brought forth key components of beer to the beer brewing industry. The sequencing and selection of a better yeast, barley, wheat, hops, and even bacteria have paved the way to brewing select beer types. In the next few slides, I will highlight the importance of genomics and the science behind the brewing industry through a case study. A German scientist, Dr. Kevin Verstreppen, is one of the scientific leaders in the beer brewing industry. He initially worked in South Africa wine industry. Dr. Verstreppen then went to get his PhD from the beer laboratory at the University of Leuven in Belgium in 1999. Most of his doctoral research at the time involved troubleshooting the problems of brewers. DNA sequencing can be done to diagnose a brew that has gone bad. You can identify the different types of bacteria in the brew. He then moved to the Whitehead Institute in Cambridge, Massachusetts to study yeast genetics. And he also discovered at that time that a repeat number in a given gene that moves to the surface of the yeast cell is involved in the stickiness. The stickiness has medical implications, and it's also important in beer. Yeast stickiness, or yeast flocculation, varies between brewing strains and influences the flavor, clarity, and alcohol content of the beer. Yeast with increased flocculation tend to clump together and fall out of solution. After Dr. Verstreppen's his doctoral work, he then went on to teach genetics at Harvard. In 2009, he returned to Belgium to study yeast and beer in the beer and wine industries. He also worked for the Swiss chocolate company, combining flavorful yeasts into chocolate. From there, he moved to his lager lab at the University of Leuven in Belgium, where he resides now. His lab quickly got involved in high-throughput mass yeast mating, genotyping, and phenotyping. The lab developed microfluidic chips that can brew up to 2,000 different yeasts at a time in picoliter batches, each containing a single yeast cell. From there, they can test the alcohol content, and in the future, they're going to be able to test aroma compounds. RNA sequencing can be done to measure the expression of the aroma compounds, and DNA sequencing can be done for genotyping, to confirm the presence and the copy number of the genes that produce the aroma compounds. While breeding can enhance a flavor tenfold, genetic modifications and genetic engineering can increase a flavor by 1,000-fold, and the Verstreppen lab has done this. Gene editing and genotyping resulted in a beer that tastes more like a banana milkshake. While interest in genetically modified beer is on the rise and the research is solid, not all of the results are published due to non-disclosure agreements. In order to fully understand the yeast genome and phenome, Dr. Verstreppen and an Illumina senior scientist and lab manager, Brian Steffi, along with an international group of scientists, beer makers, and yeast producers, partnered to sequence and phenotype 157 yeast strains. The yeast strains include 102 beer strains, 19 wine strains, 11 spirit strains, 7 sake strains, and 18 other strains. Whole genome sequencing was performed to a medium coverage of 135x on the Illumina HiSeq 2000 or the HiSeq 2500. The genomes were individually assembled by de novo assembly, annotated, and extensively phenotyped. Their research showed that the extensive process of backslopping, which is a process whereby the yeast from a previous brew is used over and over and over again for future brews, resulted in strong domestication. This domestication selects for wort sugar utilization, increases stress tolerance because, of course, the yeast are under stressful conditions, selects for flavor production, and overall, the adaptation to man-made environment is ensued in the yeast, and in turn, they can't survive, in nature. In fact, this manuscript was actually published in 2016, and they showed that industrial yeast are genetically and phenotypically distinct from wild-type strains, and they stem from only a limited set of ancestral strains, as shown here in this dendrogram. Phenotypes of domesticated strains include increased aneuploidy, copy number. This copy number variant can actually lead to increased maltose utilization. There's also an increase in genome decay. So there's a lot of there's a loss of genes that are actually involved for living in nature, and this leads to a reduction in overall survival in nature. There's also reduced sexual reproduction, and there's a reduction in all flavors. After whole genome sequencing and de novo assembly of each of the genomes, a maximum likelihood phylogenetic tree was created based on codon alignments from 2020 single copy nuclear genes shared by each of the 157 isolates. As shown here in the phylogenetic tree, yeast are diversified into 5 clades, 1 including the Asian strains, such as sake yeast, 1 mostly containing wine yeast, and a mixed clade that contains bread and other yeast, and 2 separate families of beer yeast. While most clades lack strong geographical substructure, one of the beer clades contains geographically isolated subgroups of strains used in continental Europe, Belgium, Germany, the United Kingdom, and the US. Principal component analysis shows that clustering, the same type of clustering that's depicted in panel A. Panel B shows the population structure of the yeast strains. The vertical axis shows the fractional representation of resolved populations. In the same study, the robotics used to cross yeast strains and characterize the yeast strains based on ethanol production, copper tolerance, sulfate tolerance, and maltreose fermentation. On the y-axis, you'll see our arbitrary units are AU. Ultimately, the Dr. Verstreppen laboratory is using this data to create a yeast roadmap. With proper genotyping, phenotyping, and gene expression analysis, they will be able to offer brewers at large a means to mix and match yeast to create the perfect beer or the specific beer that they're looking for. I will now discuss other successes in the genomics of beer industry. Advances in DNA synthesis, synthetic biology, and DNA sequencing have allowed scientists to engineer viruses, biochemical pathways, and assemble bacterial genomes. Recently, a group of scientists from around the world joined the Synthetic Yeast 2.0 project, and for the first time, an entire eukaryotic chromosome was synthesized from scratch. Synthesis of the yeast chromosome 3 involved the removal of various elements, including subtelomeric regions, but opened the door to the yeast genetic engineering for broad purposes involving synthetic genomics. Due to synthetic genomics, the DNA synthesis, yeast is now used in the production of biofuels, vaccines, food, enzymes, pharmaceuticals, wine, and of course, beer. A synthetic engineered wine strain was engineered by gene editing using CRISPR-Cas9, and for the de novo biosynthesis of a popular raspberry ketone aroma compound. Next, I will discuss how the genomics of barley, hops, wheat, and bacteria have contributed to the craft beer brewing industry. Genomics, including whole genome sequencing, RNA sequencing, and Sanger sequencing, have provided insights into over 39,000 barley genes. Genomes and DNA sequencing have identified an array of alpha amylase genes involved in breaking down complex sugars into simple sugars that are easily metabolized by the yeast. Genetic engineering led to the incorporation of these amylases in the barley strains and other grains, and this allows brewers to subvert the mashing process. Genomics of hops have resulted in a draft genome and a draft transcriptome assemblies. Hopbase.org was created to offer hop support to brewers and other broad applications that include medicine. PacBio, or long-read sequencing, was very useful and instrumental in defining genes responsible for the synthesis of gluten compounds in wheat. So imagine, we can have gluten-free wheat beer. Whole genome sequencing of the beer spoiling L. casei strain, or Lactobacillus casei strain of bacteria, was sequenced using the Illumina HiSeq, and the genome was assembled by de novo assembly. Bacterial genomics can be used to detect undesirable and desirable strains in beer. For example, while Lactobacillus may elicit desirable traits, it can also confer spoiling. In fact, there are many different species that include L. brevis, L. lindneri, and L. bruchecki. Lactobacillus may be responsible for off flavors. It's also incorporated in Belgian sours, lambics, American sour beers. They all celebrate Lactobacillus. A tinge of lactic acid is welcome to many and undesired by some. So now I'm going to talk about a little bit of the future of the genomics of beer. So when considering the future of the genomics of beer, you can consider that we can enhance gene duplications for the utilization of maltriose. We can omit certain types of compounds like 4-VG that confer a clove or a smoke flavor that tends to be quite undesirable. Labs are aggressively using hybridization to make other types of yeast strains and to combine the genetics naturally. They're also looking for high alcohol tolerance, drought-resistant strains. There's many different gene editing tools that can be used in order to experiment just to determine what is interesting. So once they've defined a certain gene that contributes to the flavor or the actual pleasure of drinking the beer, they can search for a strain that actually has that gene, and then they can cross the strains to make sure that they can actually incorporate that beer into their brew. Of course, sequencing is involved in every step of the way. So next generation sequencing can be used to determine how the expression level of multiple different genes, and eventually, very near in the future, and even now, there are a lot of tools out there that we can use in order to figure out what beer we're going to craft and what we're going to include in the beer. Metagenomics can be used, 16S or even 18S metagenomics, to sequence the 16S ribosomal gene, ribosomal RNA, to figure out exactly what types of bacteria are in the beer, or 18S sequencing can be done to figure out what yeast or any eukaryotic organism is in the beer. I will next move on to talk about resources for brewing, and that will complete my presentation. So the White Lab has the largest beer yeast repository in the world, and you can reach the White Labs at whitelabs.com. The Beer Advocate is a World of Beer website and magazine. It's a great resource. Ratebeer.com is responsible for, it's where customers go to find how beers are rated. Don't worry if you don't see your beer on the list. If they haven't tasted it, they haven't rated it yet. So I'm not exactly sure how they pick which beers they taste, but it, of course, is not going to be all-inclusive. Craftbeer.com is an educational craft beer website that's very comprehensive. Howtobrew.com is John Palmer's website on how to brew. He has books and a lot of fundamentals of brewing. Untapped.com is an application to document, share, and rate beers that you try. So if you travel to a different country and you've tried a bunch of beers, you may forget about what you tried. You can always look at the app to find out what you tasted on that particular day. Hopbase.org is everything hops. Thank you so much for taking the time to join me today to explore the world of beer and the world of genomics in beer. Have a great day and best of luck in your next brew.
Okay, that was our presentation. Now we're going to do a quick Q&A session. So if you have any questions, feel free to send them our way, and we'll try and get to them. We can also email you some answers in case we don't get to them. You can also email us, ngs@genewiz.com. To start, so What brewing companies are using GENOM's next generation sequencing and Sanger sequencing?
Oh, that's a good question. There's a number of different brewing companies across the country and throughout the world that are using next generation sequencing and Sanger sequencing to monitor their brews. We have a number of companies that are actually even sequencing with us, but I can't name the specific companies, but they're spread globally.
What sequencing platforms can be used for WGS, and how much does it cost?
Well, so for whole genome sequencing of yeast, the MiSeq is a perfect platform. It's a relatively small genome, so you can use the MiSeq. At GENOMX, we have the MiSeq, the HiSeq 2500, the HiSeq 4000. We have the HiSeq X10, HiSeq NovaSeq. We also have the PacBio sequel. But the MiSeq should be perfect for whole genome sequencing or also 16S ribosomal profiling for either bacteria or fungus.
Okay. How does one determine the IBU of a beer?
Oh, so that's the International Bitterness Units. People who brew, they take into consideration the number of different hop varieties that are included in the brew. So there's a wonderful website called beerfriend.com, friend of beer, that's online, and a brewer's friend, brewersfriend.com, where you can enter in the ounces of hops that are used, and then it also takes into consideration the amount of hop-emitting alpha acids that are included in the hop variety, and the amount of time included in the boiling process. And that will determine the amount of IBUs, or international bitterness units, that the beer is expected to flavor.
How do you determine the ABV of a beer?
So you can actually consider the starting specific gravity and the ending specific gravity of the beer. You can subtract the two numbers and then multiply it by a conversion factor, and that'll give you the alcohol per volume.
Do you have any information about Beer Decoded?
Oh, so that's a good question. That actually is a group that I came across during my research. Beer Decoded is a group of scientists and also just brewers, scientists and non-scientists that have come together, and they're actually looking to sequence 1,000 different beers. They're using next generation sequencing, whole genome sequencing. And thus far, they've had funding from Kickstarter, and they sequenced the yeast of 39 beers. They had some really interesting findings, actually. I just wanted to mention the group is actually led by Gianpaolo Rondo, and it's out of Switzerland. But they sequenced 39 different beers, and they found that there was one beer that had 24 different varieties of yeast in it. They were really surprised to find the number of different beers that had wild strains of yeast in the brew. Of course, that can come from a number of different areas. It could come from whatever type of whatever they're adding to the beer. I mean, I think there was one that was a cranberry beer, and it had a lot of wild type strains of yeast. But either way, this group is actually looking to sequence 1,000 different beers to characterize the entire microbial population and of the beer, doing metagenomics and also just mostly whole genome sequencing at this time.
Great. Is the gluten-free wheat currently in use? If so, how does it compare to regular wheat brews in flavor and alcohol content?
Oh, that's a very good question. So right now, what they've been doing is selecting for different wheat strains through breeding, and so there actually are brews out there that are that are more amenable to people who are have gluten intolerance. I don't know the specific brews, but to my knowledge, they're not yet using genetic engineering in crafting the beers. Or I actually don't know of any beers that are using genetic engineering. Yeah, so there are actually, it can, of course, can be done, and you can definitely craft a gluten-free beer.
Mm-hmm.
If anybody, if anyone in the audience has anything to add, you can definitely send an email to ngs@juniors.com. And if you have anything to add to the presentation, that would be wonderful. There may be some brewers out there in the audience, I would imagine.
Do you think the beer industry is ready to adopt CRISPR-Cas9 edited yeast in commercial fermentation?
Yeah, so that's kind of a loaded question. There's a lot of pushback for using genetic engineering and GMO, the whole GMO pushback that's out there right now globally. Right now, what people are doing in the Verstraeten lab is specifically just breeding the different yeast to select for specific types of specific genes and specific flavors, and they're continuing with multiple different experiments to get the correct different types of yeast and then characterize them. But right now, I don't quite think we're ready for it in the beer industry. There is definitely a lot of pushback, but I do think it's desirable by many, especially individuals who have allergies, such as gluten. So we'll see what's to come.
Definitely. How does long-read sequencing improve plant genome assemblies compared to short-read sequencing?
Oh, well, long-read sequencing is indispensable for sequencing stretches across regions of high repetitive regions. And if there's the ploidy of various grains, could be pretty high. And so it would be very advantageous for sequencing any types of difficult genomes, especially if there's long repetitive regions. Or short read sequencing cannot possibly assemble a long repetitive region. So it'd be indispensable for that.
For gluten-free beers, since they're not genetically engineered, does it mean ingredients like rice malt can help?
Yeah, it's a possibility. So what scientists are actually doing is they're looking to see, they assess all of the genes that contribute to the gluten sensitivity, and they can actually harvest strains that actually lack those specific genes, and certainly genetic engineering can help, but right now, rice type beers can actually help with gluten tolerance.
Okay.
Yeah.
And we only have one more question. What organism causes the sour flavor of sour beers?
Yeah, that's a very good question. So the taste of beer is contributed in large by the yeast. There are certain yeast strains that contribute the sour flavor. There's also several different types of bacterial strains that can also give a little bit of a sour tinge to beer. Yeah, and so I guess it depends on the yeast and bacterial strains alike.
Okay.
And of course, you can always add citrus, as a source for yeast metabolism. You can add lemon, whatever you like to. You can add fruits.
Great. If we don't have any more questions, it looks like it's going to wrap up our Genomics of Beer webinar. I hope you enjoyed the presentation, and thank you so much for participating. And we'll have this available for you to view at your leisure. Thank you so much.